When we go through and we talk about osmosis, okay, so that's anytime our water is moving. We have three words when we talk about osmosis, and those three words are hypertonic, isotonic, and hypotonic. And our hyper is if you use the term hyper breaking it down just means elevated. Okay, so you have hyperactive, right? That's somebody who has an extra active. Hypertension for blood pressure is high blood pressure. Okay, so hyper just means an elevated amount. When we say tonic, that tonic root word deals with the concentration of stuff. Now, guys, these words inside of a cell. Almost every cell is 70% water, 30% stuff. Okay, there's some variation from species to species, but not a lot. It's pretty much 70% water, 30% stuff. Most organisms are 70% water. So when we look at this, these solution names, they're referring to the solution outside the cell. A hypertonic cell, more concentrated than the actual cell. There's extra stuff out there. A isotonic, iso, think of that prefix like isometrics or an isosceles triangle. That's even. Okay, so it's an equal amount inside and outside of the cell. And finally, we have hypo. Think like hypothermia or hypotoxic, right? Hypoxic. This is our low levels. Hypotonic is where we have less stuff than inside of the cell. Are we good with these three words?
Yeah.
Kind of. All right, so guys, these three words are going to be specifically what our foldable is on. Because these are the three that often get mixed up. So did everybody get a little sheet and a big sheet? Okay, so I need you to get your little sheets and a big sheet. Now, guys, while you're getting them with your little sheet, I would like you to cut it apart into the sixth picture. You do not need to fussy cut around the cells like it just squares it from.
Oh, not my fault. I didn't think of it. I know, but yeah, if you want to, I need a small one. Why are you looking like that? This bigger?
Yeah.
What are we supposed to cut?
We're just cutting the sixth picture now.
Okay, thank you.
So, guys, the small guys. So the small pictures, we're going to cut them apart into the six pictures. That way you don't have to try and draw cells. I found you pictures of a cell. We're going to cut and paste them in. So you didn't have to draw them. But these are our options here. Okay, so we have our six little cells. Now, guys, these cells, once you have them cut out, they do not need to be like cut out next to the cell. Just squares is fine. Just put them next to you. There should be 5 per bit. So, now, Did everybody pick up this paper? Yes?
Whoa.
All right, guys, can we stop? Well, considering there are 5, which is how many people are supposed to be at a table? There was 5 per table.
It's
You did too.
I have 8 at that table, so we have 8 orders.
Uh-oh. Jamie.
I'm not I was just saying that you're arguing over supplies when you're not where you're supposed to be. I did 5 per table so that it was evenly spread.
No, I'm not. I don't know, you said you are.
Alright, okay, so once you have your 6 blocks cut out, what I'm gonna have you do is I'm gonna have you go ahead and get this piece of paper. We are going to fold where the writing is towards the center so that it makes our words. So at the top, it is going to read hypertonic. The middle is going to read isotonic, and the bottom is going to read hypotonic. Now, because the copier does not copy all the way to the edge, you will notice that in the center, it kind of cuts some of the letters a little bit. So if you would like to fill in where that letter was missed, you may. So we just folded the 2 sides into the center.
Why are you being?
Huh? The 2 sides into the center. So you do the half fold, that's your middle, you just fold each side into that line. Now, guys, ladies, gentlemen, the amount I'm having to talk over you is getting excessive. Guys, come on. If we were paying attention, we would know.
Seems too loud.
I understand, as a table, it is too loud.
What's your problem?
Not loud. So right now, we should have this. This is as far as we got. Are we all here? Do we have the 2 sides folded in? So that it looks like that.
It isn't even me that good.
Guys, can we stop? Now, at each one, now down at hypotonic, where the lines are, they did get cut off by the copier. You should see above hypertonic, we have a blank stuff, blank water. Right below isotonic, blank stuff, blank water. Right below hypotonic, can you add a blank stuff, blank water? Because it got cut off. Just like it's here and here, just the same. We just need a blank. It got cut off by the copier. That 2 of them have it.
Please report to the main office.
It's only the hypotonic. It's the only one that got cut off.
Please report to the main office at this time for
Like,
early dismissal.
Blank stuff, blank water, right? On the top, hypertonic has it. Isotonic has it. Hypotonic was missing it. So if you can add it, they do. Yes. Looking at it, right there. Yes. Right, none of the hypotronics have, it. That's why I said take a moment and write like stuff, like water. Like that's it, because adding was like, it just got cut off there. All right, so we should have our foldable folded and we wrote blank stuff, blank water. Now, we have our concentrations when we talk about water. The direction of osmosis is determined by comparing the amount of particles or solutes to the concentration. So if we start at the top, hypertonic, anytime we say something is hypertonic, it means there is more stuff, less water. Now, guys, it doesn't mean, oh, if I have more stuff than water in a solution. We are comparing it to the cell. So, for example, if a normal cell is 70% water, 30% stuff, if I have 40% stuff, it would be hypertonic because 40% is more than 30%. Does this make sense? So if our amount of stuff we have is more than inside the cell, it's hypertonic. Are we good with this idea? All right, in the center, isotonic. Isotonic is equal stuff and equal water. Now, guys, this does not mean 50% stuff, 50% water. Remember, we're comparing it to something else. If I compare it to a cell, my cell is usually 70% water. If I have a solution that's 70% water, they're equal. Right? Inside and outside are equal. That makes it isotonic. Are we good with this idea? So if my solution is equal stuff and equal water to the cell, it's isotonic. My last one is hypotonic. Hypotonic is less stuff and more water. Okay? Now, guys, can we see how any solution that exists, if I compare it to a cell, it's going to be one of these three categories. We either have more stuff, the same amount of stuff, or less stuff than what I'm comparing it to. Are we good with this? So these are the only options. Hyper, iso, or hypo. We only use these words when we're comparing two different sides, right? Usually it's a liquid to inside a cell. Sometimes you have like two different liquids, but you have to be comparing. If I just say more, guys, if I say I have more lead in my water, right? Could I say I have more lead in my water than I want and have it be like 0.001%? Yeah? How much lead do you want in your water? None. So if I have 0.001% lead, is that more than what you want? Yeah. It's not a lot, but it's still more than what you want, right? Same way as if I could go through and be like, all right, well, I have less money than I want, right? I can have a lot of money. Does that mean that I can't have less than what I want? Right? You can, right? Because you could keep going up, right? So when we use those terms, they're comparative terms. Are we good with this idea? We can only use them when we're comparing. All right, do we all have the outside done? All right. I'm gonna have you open it up. We are going to go across the top. It's hypertonic. So water goes to where there are more particles. In hypertonic, we had more stuff than water. The empty space in the picture on the slide is water. The dots are stuff. Now, when it's hypertonic, above strength, the concentration outside of the cell is greater than inside. This means more water is going to leave because the water is going to go from in here where there's a lot of it out. Does this make sense? Now, let's go ahead and look at our line. So on our animal cell, which way is the water going to move? In, out, or both? Out. So our water is going to move out of the cell. That's for both the plant and the animal. The water is going to leave the cell. Yep, both animals and plants are both going to have the water leave. Well, now let's think about our cells, right? Our animal cell just has a cell membrane. When water leaves the animal cell, what is the animal cell going to do? Yeah, it's going to shrink or shrivel, right? Imagine a water balloon that has a leak, right? It's going to shrink and shrivel up. So right here on the animal cell side, we are going to put the cell shrivels up. Now, I want you to take a moment and look at your pictures. You have 3 of the cutout pictures that were squares. Those were your plant cells. And we have 3 that were not. They were kind of ovally or circly. Which one looks like it's a cell that's starting to shrivel? Shrink. Yep. Yeah. So it's the one that looks all like wobbly. Yeah, it's getting all the water left in. Guys, that's the one I'm going to have you take right here next to our animal cell, because the animal cell shrunk or shriveled up. Or you can tape or glue, either one. You just want it to be attached. So it's the one where it looks like our animal cell has like the water left it, like think of a water balloon that lost some of its water. Yeah. And you're just gonna put it in the center next to the animal cell. All right, so now let's think about our plant cell. If the water is leaving the plant cell have a cell wall. So can the cell shrivel up? No, there's a cell wall in the way. So guys, what we're going to see in a plant cell is the cell membrane is going to actually, the cell is going to pull away from the cell wall.
Yeah,
Yeah, in the bin. Now, if you look at your plant cells, those are the square ones. You want to find the one where it looks like it's pulling away from the cell wall. Now, guys, if this happens too much, if it pulls away too far, the actual cell membrane is going to tear and the cell will die. This is why if you don't water your plants, the plants die. Okay? So, for hypertonic, the water moves out. For our animal cell, it's going to move out and the cell is going to shrivel up. For the plant cell, the water is going to move out, but what we're going to see is the cell membrane pulling away from the cell wall. It's gonna pull away from the cell wall. Everything inside, because it's gonna try and shrink, but it's attached to the cell wall, so it ends up looking like that. So guys, do plants do real well if they don't get water? No, they do not handle dehydration well. Plants can't get dried out very well. They get real wilty and not do real well. All right, so that is our hypertonic. Yep. The cell membrane pulls away from the cell wall. So it pulls it all away. And we use this. All right, if we look at our isotonic means same strength. Guys, for isotonic, it's equal inside and outside, and the cells try to maintain their homeostasis, right? Or the same level. Now, guys, with our water going in and our water leaving are going to be equal. It doesn't mean that it suddenly became impervious. It just means the amount of water going in and out stay the same. Okay, so it's in and out. So for both our animal and our plant cell, where it says water moves, you're going to put in and out on both of them. Now, if our water is evenly moving in and out, is the cell going to change? No, so both of them are cells going to stay the same. Yeah. So now I want you to look at our pictures here. So for our animal cell, it is going to be the oval that looks like our nice picture when we draw our animal cells. It's a nice oval, all evenly shaded. That's going to go for our animal cell. A nice even oval. Yeah, it's an animal cell, right? It's not a plant cell.
No, it's not.
Remember, plant cells have a cell wall. Those are just rectangular ones. It's all right. Oh, wait.
That's an animal. The animal stuff. Rock one is the plant. Yeah, the circle.
I haven't even got that one yet.
It's good. It's over there, but it's not.
All right, for the plant cell, guys, you have two plant cells remaining. They're the squares. One of them looks all swollen and puffy. The other one is the normal plant cell. So we want to put our normal plant cell over here on the side for plants. That's the one that's not swollen.
How do you know it's not swollen?
Well, if you look at one of them looks like it's puffy. That's the normal one, yeah.
So the normal one?
That one's normal.
Which one is not?
The normal. So it doesn't stay, everything stays the same. Oh, wow.
Oh.
The other square one looks like it's puffy. That's gonna go across. So if you look at the two squares, other thing that's something. That's very cool.
Oh, wait, that's gone.
So right now, we should have hypertonic and isotonic. Hypertonic and isotonic. We have one more.
Finished.
All right, are we ready for hypotonic? All right, guys, so if we have hypotonic is our below strength concentration. It means the concentration outside of the cell is less than inside. That means there's more water outside than inside. When this happens, more water enters than leaves, and so the cell will swell and possibly burst. So the water is going to go in. Now, guys, can we stop the side conversation? Irrelevant. Guys, if I want you to take and picture that water balloon again, right? So our animal cell's kind of like that water balloon. What is going to happen to a water balloon if we just keep adding water to it? It's going to get bigger and bigger and bigger, and then it's going to pop. So our animal cell is going to swell, then burst. Well, like get bigger, or get bigger and then burst. Now, guys, if you look at your pictures, look at your animal cell. Do you see the one that looks like it's popping? That is what happens to our animal cell. It swells up and bursts. Now, our plant cell, guys. Ladies, gentlemen, come on. Yeah. Nope, it's dead. It died. Now, guys, an example of cell swelling and then bursting that you may have had encounters with is diaper rash. What's happening with diaper rash is baby skin is so thin, it doesn't have a lot of layers. If you do not change their diaper really quickly and they have the moisture of their pee, guys, can you stop the side conversation? Please. Talking over top of you guys is really irritating. Guys, if you have the babies don't have like really strong pee, right? It's mostly water. If you do not change their diaper really quickly, what happens is their cells of their skin actually pull in the water from the pee until they pop. And you literally have the skin has basically dissolved because the cells all pop. And you end up, if you have ever seen diaper rash, it's basically like a puff blister. It's like little blisters that have popped. That's what happens in diaper rashes. Those cells had swelled up until they popped. All right, so let's think about our plant cell. Guys, our plant cell has a cell wall. So is it going to be able to swell to the point that it pops? No. is it going to be able to swell up some?
Yes.
Yeah, so our plant cell is only going to swell up. It will not pop because it has the cell wall keeping it from popping. Okay, plants can deal with too much water a lot better than animals. Animals can deal with it getting dehydrated a lot better than plants. Does this make sense? So for our plant cell, it's the water moves into the cell and the cell just swells up. And so that's going to be our last picture, is that cell swelling up. Now, guys, this is your second foldable. This is the two foldables you need for your quiz, with the cell membrane foldable we did with all the colors, and this one. Not the 14th, 15th, the 14th grade. Next class, because I have them, they're in format, they're just, I don't want to send them to you. I still have two people. I'm trying to make it so they have so they still get. But basically, if they don't make it by next time, then they don't only get to watch. They only get the minute. All right, so guys, I'm going to ask, the easiest way to do this is the back of your foldable is nice and blank. You can go ahead and glue that down in your journals so that you don't misplace them. There's a glue stick and a glue bottle for every color. There.
Yeah, share.
There.
He's pointing at me over and over. Speak up.
Yeah.
You can.
What? I know, man. My oldest bedroom was like never really went all the way. The other one did. Here. Very successful. Did I enjoy all of these? No. Other my oldest is 28. I have 28 for each. My youngest one. My oldest, I have two older, for 28 and 26, and I have 10. All right, are we all good? We have our full flow. So guys, next week, your quiz is your cell membrane transport foldable and your osmosis foldable. These are the two on the quiz. Huh? We're doing them next class. I've been waiting for, I've had just a handful of people that just have been out and out and out. And I was trying to make sure they had two chances, but they're kind of short now. right. What I now need you guys to do is to go ahead and log into your Gizmo account. And you have misplaced your password. I have them. So guys. Yep. Do you have one? Do you remember what it is? Then you can use that. Oh, wait, so I have to get you added. Hold on. So I have to add you to the class? Huh? You are 831. 831.
What's the username again?
Huh? The username is Vegabio. The passwords are up here. Guys, you should not be logging in with a username and password. You are on the wrong one then. You want to log in with just the username Vegabio. You want to pick class 831, then click on your name.
Wow.
831 is the last 3 digits of our class. Can we not? It's Vegabio is the.
Caption it.
It will log in either, but I did a capital V and a capital B, but it should let you in either way.
We have to log into this.
Yes,
Just say I just mine.
you have not yet.
Oh, it's.
The new one.
Oh, no, that's.
Oh, I don't know. I'd have to go back. I didn't check it after.
Are you in a Zoom call?
Huh? I'm recording for the classes that I. How would you not? All right, so.
We are considerably lucky.
The. So I have to trying to record. It's one of our classes, we're trying to find some minutes. They're one day. Giving them a recording of what we- 831. Nope, they put you in. Do you need your password? Okay. If so, let me know, because they moved you, the county moved it. I don't have it on my printout, but I do have it right here if you want to see it. What it's not working. So did you put in Vegabio? And then you picked your class? What do you mean?
Oh.
Yeah. Right. I gave you a little bit. I still not to this day. To this day. I still do my mom to this day. Yeah. Like, you know how bad that would have been. Yeah. How you, bro? I'm not like a slide. You know.
Guys, what we are going to go through and do is you are going to be doing come on. We are in class 831. What are you looking for? Your username is Vegabio.
Wait.
Yeah. Yeah.
When are we going to bring you guys?
I can do that once I get you guys started. If you guys started. All right, so guys, in your class, ladies, gentlemen, come on. In your class, you want to find the brain. It looks says osmosis right down here. That is the one you want. You're going to tell it to launch. Now, the high school is the only one assigned to you. Yeah, you don't need the AP level. It was just the high school level. So guys, this one is very similar. This is another vet case study. This is an actual organism as you go through, just like with Clara the dog, that they end up okay. All right. So at the end, we are successful at saving the baby calf. So you are a veterinarian. There's a 5-day-old calf named Clark brought to your clinic. They're bovine. They're around 110 pounds at 5 days old. Guys, come on. Now, they sent this video of the calf having its seizures. It is real. Now, Clark also had diarrhea and was given 8 quarts of water in addition to the normal bottle milk. It is going to make you open up the glossary. Osmosis. Yes. It's not a username and password. You are trying to sign in as the teacher. You need to go. You're not an educator. You're going to be in the student. You have to log in with my. Just bigger. Yep. Now, your goal is to find out what is causing the seizures and pick the best treatments. So we're going to learn about the seizures and osmosis. We're going to use our arrows. Yes, it was having a seizure. So seizures may occur when too much water moves by osmosis into the brain. Osmosis is the diffusion of water across a membrane. Not always, that's one of the ways. To understand osmosis, you must first understand diffusion. Diffusion is the movement of ions or molecules from an area of high concentration to low concentration. Guys, I don't mind if you chat, but you have to do it at full volume. So if we go through, if we diffuse, this is what happens over time. It's not that any one molecule stops moving, but over time they spread out. This is just the water diffusing. This is what happens with diffusion. Huh? This is what happens anytime we do diffusion. So they're starting out at the very simplest, and then we're going to get into how it gets into the brain. Both. Yes. Yes. So let's learn about our cell membranes. What happens if a membrane is added? Some molecules, like oxygen, can diffuse across the cell membrane. Otherwise, where the membrane is permeable to oxygen. So if we look here, we can see that they actually go through the membrane. The difference in concentration across the cell membrane is called a concentration gradient. Molecules diffuse down their concentration gradient from an area of high concentration to low concentration. So create a concentration gradient to make the oxygen molecules diffuse across the membrane into the cell. So we have on the outside of the cell, do we want it to be 0 or 30? And then inside of the cell, what do we want? Now, if I have 30 on the outside and 0 on the inside, there was more on the outside, so it makes it go into it because it wants to end up leveling it out. Does this make sense? After it's done, they'll both be right around 15-15. We're good? Can all substances diffuse across cell membranes? Let's try it with sodium. This membrane is impermeable to the sodium ion, so they cannot go across that membrane. What about water? As you have seen, not everything can diffuse across a cell membrane, even if there's a concentration gradient. So what about water? So if you watch that animation, water molecules cannot easily diffuse across cell membranes. So cells need a way to facilitate or help the diffusion of water across their membrane. What if there were pores or holes in the membrane? We now have those pores. There should be a the cap.
Oh, yeah, it was
Yeah.
black.
Yes. I was totally confused.
That's what happened.
This is in the handbook. So we're going through this and then it's gonna go back to the handbook. Okay? All right. The pores made it possible for the water molecules to make it across the membrane. This process is called facilitated diffusion. So guys, are those just regular holes? What are they made out of? We talked about it on our cell membrane foldable. It's in the membrane. It's not just the hole in the lipids, otherwise it would close up. What's actually there? The channels, right? Those the pores in the cell membrane that help the diffusion of water are called aquaporons, which means, guys, which means water pores. Aquaporons only allow water molecules to move through them. And you can see now we have the protein animations there. The green proteins, because it's not actually a big gaping hole. It's a special protein that just lets water through. When the number of water molecules moving into and out of the cell are equal, it's called an equilibrium. Because cell membranes are permeable to some molecules, like oxygen and not others, such as sodium, they are called semi-permeable. Ladies. Or selectively permeable membranes. Like other molecules, the water molecules diffuse from areas of high concentration to low concentration. When this diffusion of water happens across a membrane, it's called osmosis. How does solutes affect osmosis? A solute is a substance that's dissolved in a liquid. Sodium is an important solute in the body and usually enters the body as salt or sodium chloride. So when that salt dissolves, this is what it ends up looking like, is those water molecules wrap around each individual atom and pull it apart. It's fine. You're good. The water molecules bind and attach to the sodium chloride that separates them. Solute-bound water molecules cannot pass through an occuporon. Only the free water can pass through. So if it's got something dissolved in it, it can't make it through. Only pure water can. So, what would you predict would happen to the concentration of free water if I add salt to the solution? So if I drop in some salt, the salt binds, the water binds to the salt, and we end up decreasing the amount of just loose water. Now, can you predict which way the free water molecules will diffuse? Outside of the cell, we have 6 solutes and 12 free water. Inside, we have 3 solutes and 32 free water. Which way is the water going to go? See what happens? Our free water does move out of the cell. It moved down their concentration gradient from an area of high concentration to low. Guys, key concept, the side with the higher concentration of sodium ends up with less free water. Does this make sense? So the more stuff you have, the less water you have space for. Solute concentrations across the membrane. In the last example, the solute concentration was higher outside than inside. This solution is called hypertonic when compared to the solution inside. Hyper meaning more. Here, our solution is hypotonic when compared to the solution outside. Hypo meaning less. In this example, the solute concentration is higher on the inside than the outside. The inside is now hypertonic and the outside would be hypotonic. When the solute concentrations on both sides are equal, the solution is called isotonic. What would happen to red blood cells if they were placed in hypertonic? You can use your foldable. Good trick. Good call. They would shrink. The hypertonic has lower concentration of free water, so the water diffused out of the cell. What about isotonic? Correct. The isotonic solution has the same concentration of free water as cells, so the cells stay the same size. And hypotonic? Correct. The hypotonic solution has a higher concentration of free water, so the water diffused into the cells and the cells swell. And guys, those of you who want to work ahead, are welcome to. I was just walking through all of them in case you had not. Solute concentrations are not only important for red blood cells, but also the organs of the body like the brain. Nutrients for the brain are delivered via blood vessels. The wall of the blood vessel are semi-permeable to allow free water to move between the blood and the matrix of the brain. In the image, it's the blood hypertonic, isotonic, or hypotonic compared to the matrix of the brain. So the solutes are the green. What do we think? What does the blood look like? Correct. It is hypotonic because it has less solutes in the matrix. If you look at the brain has a lot more green dots, which means the blood is hypotonic. It had less green dots. It was less stuff. Are we good? Which way will the water move? So it is going to be from our blood to the matrix because it wants to move towards the extra stuff. Right? So hypo, it moves out of hypo. So there's more free water molecules in the blood, so the water is going to diffuse into the matrix of the brain. Because the brain is inside of the skull, the brain can only swell a small amount. What would happen to the pressure in the brain if the water moves into it from the blood? Yeah, so if we add water to our brain, it's going to swell and the pressure is going to increase. Huh? That's one of the ways that seizures can happen. It's not all of it, it's one variety. So as water moves into the brain from the blood vessels, the pressure increases because the skull limits how much the brain can expand. Ladies, you know you have to do this, right? Huh? What happens when pressure increases in the brain? So our brain contains billions of cells called neurons. These neurons control how the body works by communicating through electrical signals. The frequency of these signals is called the firing rate. If you move the slider, if we bring it low, we get less firing rate. If we bring it high, it sends signals faster. If the firing rate is abnormal, either low or high, the brain does not work normally. For example, if pressure in the brain is too high, the neurons will fire too often, and that can cause seizures. So, we've completed the handbook. If you need to review any, you can go back to any of these topics. So if we come back to our case, we're going to collect some data from Clark's brain. Come on. So we will go into the brain. This is a neuron. Click to add the neuron firing rate to the bottom of the screen. Here is a blood vessel. We're going to put our blood pressure reading. The brain matrix pressure, we're going to add that one. And the net free water movement is our last one we want to add. Now, if we zoom in close, this is what's happening in our blood. And up here is our matrix. That's our blood vessel wall. And the brain matrix. The green dots are the sodium. This is the sodium in the matrix outside the blood vessel. If we add matrix sodium, the sodium inside the vessel, we're going to add that one to our data that we collected. Yes. Huh? Yeah. We have our free water and the aquaporons. This movement of free water is an example of osmosis. So if we do our live feed, so he is actively having seizures. If we collect our sodium level, we collected the free water movement data and the sodium data, our firing rate data. Let's analyze. So we upload our data. In the matrix, the sodium level is 137. Is it low, normal, or high? The pressure is 27. It should be between 5 and 15. Blood sodium is 114. The normal is 137 to 146. Our pressure is 22. It should be between 20 and 25. And the firing rate is 4.5 instead of 2.3 to 2.5. So we look at our free water. What is going to describe the net free water movement between the blood vessel and the matrix here? What are we seeing? Into the vessel, in equilibrium, or out? Correct. Now we want to interpret. So using this information, our sodium, our pressure is high. And what you've learned about the seizure handbook, why are the free water molecules diffusing out of the blood vessel? Where do we have more salt? Matrix. So where's water going to want to go? Where there's more sodium, right? Or more solute. Do you want to put a sentence here? We're not looking for an essay. We're looking for a sentence. Why do you think the pressure in the matrix is high? There you go. Where our water is heading, right? More, if the water is constantly going in somewhere, it's going to increase that pressure. So let's put these in order. We're going to sequence our events. So the first three ones are put in for us, which was the normal blood and matrix sodium concentration. We lost sodium due to diarrhea, right? That was the thing they said started this off. They were then given too much water. What happened next? What do you guys think? The calf was given too much water. What's going to happen next? I had low blood sodium, so let's see if that one goes there. After we get low blood sodium, what's going to happen next? Yeah, our water is going to move from the blood into the brain. Once we have water going into the brain, what happens next? Our pressure is going to go up. Once the pressure goes up, what's it going to cause? Yeah, the neurons are going to fire off. And then what are we going to see? Does this make sense on that sequencing? Guys, when you're sequencing, if you're ever not sure, try to be like, okay, well, what causes that? What causes that? And you might end up with like 4 here and be like, oh, wait, that must have gone up at the top, and you can slide them all down. Right? So, guys, this is one of the reasons why if you ever have a gastro illness, throwing up, diarrhea, do they recommend you drink lots of water? No, they say fluids. They want you drinking things like Gatorade, Pedialyte, juice, soup, right? The broth, stuff that's going to have those salts. Yes. All right, so let's diagnose our patients. So, we have a cerebral edema. Too much water in our body tissues is called an edema. Edema in the brain is called a cerebral edema. It can happen if the concentration of sodium ions in the blood decreases. It can be, the brain can be damaged. The skull limits how much the brain can swell. Increases of pressure in the brain stops normal neuron function. Animals and people with cerebral edema develop changes in behavior and often have difficulty walking, become blind, or develop seizures. If not treated, cerebral edema may result in death. Epilepsy is a condition where the neurons in the brain fire too quickly. Increasing the firing rate can also lead to seizures. Common causes of epilepsy are damage to the brain, changes in the proteins that control the movement of ions across that barrier, right? So if we, instead of letting the sodium not go through, if it starts just letting the sodium go everywhere, the water follows it, and it can do that. Brain tumors and sometimes infections from bacteria. So, guys, which one do you think this was? And explain. And again, that was because it was from an excess of water. Not abnormal protein. We just did our evaluation. Were we on anti-epileptic, right? Huh? All right, guys, I am going to have you guys go ahead and pause. We can click our treatment here. We are going to finish it up when we have our downtime on our lab next class. but you should have gotten to the point where we're kind of at our diagnosis. Yeah, you gave your diagnosis of whether you saw it was edema or epilepsy. And then it's gonna go through the treatments. So it went through the anti-epileptic, was not effective. It then lets you revise if you would like to. And then the treatment is things like saline solution. And we are going to finish this next time. Good? Okay. So I'm going to jump back to here. Our exit question, guys, is how do you decide which way the water will move? Which way is it always going to want to go? Wherever there's more stuff. It doesn't have to be sodium in the examples that use sodium. Our body's sodium tends to be the one we use to move water around, but it doesn't have to be. We'll see the same thing if you have like extra sugar. That's one of the things that will also happen if you have like a lot of sugar. It will pull water out. Guys, if you have ever taken fresh fruit and sprinkle a little bit of pure sugar on top, right? Like fresh cut strawberries or like blueberries, you put sugar on top and let it sit. What does it turn into? Turns into like syrup, right? It's no longer nice fresh fruit. The water went because of osmosis. It left that fruit towards the sugar, that dry sugar that was on top. If you leave it like overnight with strawberries, like you put a little bit of dry sugar on it, the next day you're going to have strawberry syrup. And those strawberries aren't going to be like stiff. They're going to be kind of wobbly. Their water left them and went into the sugar. Another example using salt would be pickles. If you've ever done like a quick pickle of onions or cucumbers, you take them, you put a little bit of salt, seasonings, the water leaves them. It's one of the reasons why you don't ever put dressing on salads before you serve it. Otherwise, the salad all gets soggy. We're going to finish next time. Does this make sense to everybody? All right. Don't.